Unless you've been practicing extreme social distancing long before this last winter, you've probably heard of the novel coronavirus, or COVID-19. We all watched in fear and disbelief as the virus spread across the globe in a matter of months, ushering in the worst pandemic in over 100 years and infecting 10% of the world population. But what if I told you there is a respiratory virus that has infected 100% of the world population? In fact, 2% of you listening to this talk were hospitalized, and another 1% developed severe long-term effects, such as asthma and even learning deficits. Some quite traumatic events that you will never remember because you were an infant. This virus is respiratory syncytial virus, or RSV, and it is the leading cause of respiratory infections and hospitalizations in infants. There is currently no licensed vaccine available for RSV, and that's not due to a lack of trying. Scientists have worked for over 60 years to produce a vaccine, and in one instance, an experimental vaccine actually made RSV symptoms worse, resulting in the tragic deaths of 2 children. Now, vaccines are good, we all know that. They help our bodies build a bigger and better immune response against things that make us sick, but RSV is very, very good at using our immune system against us. Now, imagine your lungs as a river, and fish swimming in this river is oxygen flowing through our airways. Normally, we have a few boats guarding the rivers, removing trash and keeping the water clean. This is much like immune cells in your lungs, patrolling and removing bacteria, pollen, and debris. Now imagine RSV infects your lungs, causing a massive danger signal known as inflammation. This signal calls hundreds of boats to enter the river and begin dumping oil from their engines, like mucus in the lungs. As you can imagine, this makes it very, very difficult to breathe and can cause infants and the elderly to require hospitalization and even lead to death. Gaining a better understanding of how RSV causes inflammation and disease is absolutely critical to the development of a safe and effective vaccine. Our lab had previously noticed that 2 similar strains of RSV taken from sick children induced very different levels of disease. In utilizing genetic techniques, I was able to replace proteins from the RSV strain that causes a simple runny nose with proteins from the strain that could put an infant in the hospital, and see if we could make that wimpy strain better at making us sick. With this approach, I was able to show that as little as 1 small change in a very complex viral protein was the culprit, as well as identifying key signals within our bodies that cause the immune system to overreact and do more harm than good. This could be very useful in forecasting RSV season severity before children end up in the hospital. This also has the potential to save thousands of lives each and every year. And we sincerely hope that our contribution to the field of RSV research will serve as a stepping stone for others and make that dream a reality. Thank you.